Hello everyone, welcome to ITLS Academy. In the module of Medical Biochemistry, uh, today myself Kamini Gautam will talk about biological membranes, structure and their function. In our first lecture, we spoke about cell and its organelles, and we came across a word called cell membrane or cells which are surrounded by a membrane which is also known as the plasma membrane, which separates the cell contents from the outer environment. So in this lecture, we will review and talk about current knowledge on membranes. So this is the outline. So in this lecture, we will talk about uh, the structure and function of cell membrane. We will talk about the plasma membrane as a prototype. We will talk about the lipid composition and its types in the plasma membrane. Or we will talk about the compartments and emphasizing its role in sustaining organelle structure and function. We will talk about in detail about the proteins presence and their types in the membrane. There could be integral membrane proteins, there could be peripheral, etc. And then we will also talk about the nature of the carbohydrates which are attached to the plasma membrane. We will also discuss about the formation of lipid bilayer. We will in detail talk about the function of certain membrane components like ion channels, transporters, gap junctions, etc. We will also talk about in detail about the transport mechanisms and how the molecules transport through these membranes. So if we'll start with uh, biological membranes. What are the biological membranes? Biological membranes are the key elements which are required to maintain the cell architecture as well as its physiology. Basically defined as the cell boundaries and internal organelles in eukaryotes. Plasma membrane is a prototype cell membrane scaffold extensively, and it plays a very important role in cell-to-cell communication and the initiation of intracellular signals among other functions. It basically acts as a barrier which separates cellular contents from the environment. It is an absolute necessity for a life. Plasma membrane have a selective permeability that mediate the flow of molecules and ions into and out of the cells. It also contains a lot of molecules on their surfaces that basically provide for the cellular recognition and communication. Eukaryotic cells contain many internal membrane system which surrounds the cell organelles, and each internal membrane system is specialized to assist in the function of organelle it surrounds. Critical to this function is the plasma membrane compartmentalization in lipid microdomains, which controls the localization and productive interaction of proteins which are involved in cell signal propagation. So, in general, membranes are basically, as I've already mentioned, they separate the cellular contents from the environment, and membranes are basically composed of lipids, protein, and carbohydrate. So the major content of the major content of lipid and protein is present as compared to carbohydrate in the plasma membrane. So this table basically represents here and shows the composition of different membrane contents, lipids, protein, and carbohydrate. So these are the, as I mentioned, lipid and protein are the major component, and as you can see, their percentage is very high as compared to the carbohydrate. They're almost covering the 100% of the membrane. So these are the organelles, like we have talked about the endoplasmic reticulum, mitochondrial membrane. So these are the different membrane structure, and these are the different composition of lipid, protein, and carbohydrate. So basically, they varies widely from one type of membrane to another, but typically it contains 40% of the dry weight of its lipid, about 60% of proteins, and 1 to 10% of carbohydrates in general. And all membrane carbohydrates are covalently attached to the proteins or lipids. So basically, all three are required in a combination for a plasma membrane to act as a barrier which separates the cellular contents from the environment. So now we'll discuss in detail about each individual component. We'll start with the lipids. Lipids are the major component, and this table basically shows the composition of different membrane and the presence of various types of lipids within these membranes. So here it shows the content, the percent of dry weight, The major lipids which are found in each membrane are cholesterol, sphingomyelin, and glycolipid. Now, if we talk about lipids in detail, basically known as the basic structural components of cell membranes. They're amphipathic because if you look at it, the structure, this is basically having a head or tail structure. The head is a polar, it's hydrophilic in nature, and the tail is hydrophobic, which is non-polar in nature. That is why they are known as amphipathic in nature. Uh, Basically, so what happens is they basically depend upon the cells basically depend upon the lipids for following three main functions. First is energy storage. So what happens is there are small lipid droplets which are used for this energy storage function, which mainly contain the triacyl glycerol and sterile esters. And basically because they have a relatively reduced state, these anhydrous reservoirs are needed for their efficient storage of calorie reserves and as stores of fatty acids and components for membrane biogenesis. Another function is compartmentalization. They plays a very important role in the compartmentalization. Basically, it provides a physical barrier and it acts the physical basis for spontaneous membrane formation because of these hydrophobic tails or moieties, and they are prone to self-associate when they are basically dissolved in water. So this predisposition of an self-association enables the segregation of internal mildew from the extracellular matrix, and when the first cell originated there's a budding, tubulation, fission, fusion. All these functions are happening because of the function of these lipid molecules is a compartmentalization. It also plays a very important role in signaling. It basically plays an important role in signal transduction and messengers transformation. These are the functions which are major functions of lipids. Now, what are the types of lipids which are present in biomembrane? So there are basically, we'll start with the fatty acids. These are the major components of most membrane lipids. And basically, this is the structure of the two of the fatty acids. The non-polar tails of most membrane lipids are long-chain fatty acids, which are attached to the polar head groups, such as glycerol 3-phosphate is an example. About 50% of the fatty acid groups, they are saturated. That is, they contain no double bond. The most common saturated in fatty acid group membrane lipids, amino acid which the in the animals which contains 16 to 18 carbon atoms. Now oleic acid is the most abundant fatty acid in animal membrane. Other than you have a linoleic acid, arachidonic acid, etc. Now what happens is the 50% of fatty acid groups are saturated. The other half of the fatty acid molecule contains one or more double bonds, that is unsaturated or polyunsaturated fatty acids. The degree of unsaturation basically determines the membrane fluidity. Now we have another type of lipid which is glycerophospholipids, in which this is the another group of major component of biomembrane. So what happens here is there is a glycerol molecule with a phosphate esterified at the alpha carbon atom. Then you have a two long chain fatty acid which esterifies to the remaining carbon atom. And there is a structure of major alcohols which is esterified to phosphatidic acid to form glycerophospholipids. Then you have the examples of this glycerophospholipids are phosphatidylethanolamine, phosphatidylcholine, phosphatidylserine. The major function they plays in metastatic cancer cells, they are generally basically ether-linked lipids. Their role is suggested in invasive properties of these cells. That's why they plays a very important role in metastatic cancer cells. Now we have another which is sphingolipids. This is the most abundant in mammalian tissues. Have phosphoryl choline which is esterified at the C1 carbon. It comprises another group of lipids found in biological membranes, especially in the tissues of nervous system. There are three types of sphingolipids: sphingomyelin, cerebrocytes, and gangliosides. And about 6% of the membrane lipids of gray matter of the cells in the brain, they are gangliosides. Now there is a fourth type of lipid which is present in biomembrane is cholesterol. It is the third major lipid, compact rigid hydrophobic molecule. It is another common component of the biomembranes of animals, but not of plants and prokaryotes. It has polar hydroxyl group at C3 carbon. It is oriented with its hydrophilic polar heads exposed to water, and its hydrophobic fused ring system, which is attached hydrocarbon groups buried in the interior. It helps to regulate again the fluidity of animal membranes. Now there are a lot of so what happens is, I've already mentioned that lipid act as a energy storage. How these things happen, how lipid droplets form, what are the roles of triacyl glycerols. So there are certain properties of the lipids. There are lipid aggregates. So what happens is, so if you look at the structure, as I've already mentioned, there is a hydrophilic head and a hydrophobic tail. So this has a property to form a micelle-like structure. What happens is, if you put them in a water, they can form a surface film or a small micelle-like structure, in which the hydrophobic tail is facing inside. However, all the heads are facing the outside. The derivatives can form larger aggregates which are held together by all hydrophobic forces. Now you have a triacyl glycerols which form large spherical fat droplets in the cell cytoplasm. Now phospholipids and glycolipids, they also have a property to form self-sealing lipid bilayers, which actually act as a basis for all cellular membranes. Now, what are the lipid bilayers? You must have heard about this term, that there's a formation of a lipid bilayer. How and what happens when there's a formation of lipid bilayers? So it's basically, if you look at it, the structure, this is in the water, which is an aqueous phase. You have a polar head, you have a non-polar tail, which are facing towards each other. This is basically a sheet-like structure, which is wide across like 6 nanometer, and there are close boundaries of the cells or organelles. The hydrophobic effect and the solvent entropy provide the driving force for the formation of this lipid bilayer. These are the two key important forces which are required. A lipid bilayer can form only when the cross-sectional areas, if you look at it, the structure, hydrophobic tail and the hydrophilic polar head are about equal. Glycerophospholipids and sphingolipids fulfill this criteria and hence can form the bilayer. The lysophospholipids have only one fatty acyl group. It cannot form the bilayer as the polar heads are too large. So these are the examples of the lipid which can form and which cannot form the lipid bilayers. Now, as I've already mentioned, there is always an effect of fluidity on the membrane. So what happens? What are the effect of temperature and fluidity on the formation or not formation of lipid bilayer? So what happens is, in a lipid bilayer, there is a hydrophobic chains of the fatty acids which can be highly aligned or ordered and provide a rather stiff structure. So what happens is, sometimes there's a transition happens from a ordered, which is a crystalline or a gel-like structure, become very disordered state, which is a fluid arrangement, or you can say it's a liquid-like, which is also known as a transition temperature. Now what happens is, if you have a longer and a more saturated fatty acid chains interact more strongly with each other via their longer hydrocarbon chains, and thus they cause a higher value of transition temperature. Hence, the higher temperatures are required to further increase the fluidity of the bilayer. Now, as the membrane fluidity increases, its permeability to water increases, and other small hydrophilic molecules also increases. Lateral mobility of all the integral proteins also increases, which helps in the transport of ions and molecules. Now, what happens if you get a defect in lipid? If there is a lipid imbalances, so they are associated with a lot of human pathologies. So, the lipidome of if you call it lipidome cellular organelles is remodeled not only upon physiological conditions but also under pathological conditions. And those changes in the cell membrane, lipid composition, in turn affect the biophysical properties, of the microdomains and the ability of signal propagation by proteins and signaling lipids. For instance, there's a propagation of microdomains and the ability. So what happens is, there are several different pathologies developed with alterations in the lipid raft composition and structures. In further detail, if we want to talk about in further detail, these are the four panels. If you can see here, the cancer. So, for example, phospholipid biosynthesis gets affected. Phospholipids regulate endocytic processes which are required for propagation, and there has to be regulated. So there's a growth, immunosurveillance, synaptic transmission, cell migration, and the other functions which are happen due to the presence of these phospholipids. So when there is a defect of these phospholipids and the ratios of these phospholipids, cancer occurs. Now there are also a lot of metabolic diseases, which is for example lysosomal trans disorder, which is a lipid storage disorders. If there's a defect in the sphingomyelin and cholesterol and galactolipids, there's a metabolic disease is happening. It leads to the obesity, it leads to the diabetes, which are associated with saturated fatty acid. So there's a if you have an intake of saturated fatty acid, for example palmitic acid, it has been shown to induce of endoplasmic reticulum stress and ultimately lead to cell death. Now you have another is called neurological disorders. In most of the Alzheimer's and such kind of neurological disorder, it was found that there is a glycophospholipid, sphingomyelin, cholesterol. These are the levels which found to be increased. If you can see here arrow, this arrow represents there's a level of increase, and there's a lipid imbalance happens in certain kind of disorders. There's a immunological disorders as well, which are developed with vascular, thromboembolism you call it, and it also leads to this anti-PL syndrome and anti-PL antibodies. It can lead to the miscarriages sometimes. So there are a lot of disorders and disease which are associated with the lipid imbalances. There's one of the most common disease which is associated is known as a cystic fibrosis, which is not mentioned here, but that's the most common diseases which happen because of the lipid imbalance, which impair, the surfactant ability in the lungs, and it promotes a lot of bacterial growth. There's a lot of bacterial replicate in your lungs, which basically leads to decrease the breathing ability. So it happens because of the defect in the transporter, which transports the chloride ion, that we will talk about in detail later. Now, when there are disease, people are started finding out what are the lipid-based therapeutic avenues also. Could be a membrane lipid therapy. So it is very, very clear that many cell functions takes place in membranes and their surroundings. So there's an aim to target the membrane composition or membrane components, to modify membrane structure or compartments. That is, you can have a drug induced uh, regulation of membrane lipid composition. You can have a structure based, you can which shows higher efficacy or specificity and safety, and has become a potential alternative to conventional drugs. There are a lot of currently being developed for several medical conditions. So like for example, you have a molecular basis for the targeting the plasma membrane. So here you can have a lipid alterations which affects the protein-protein interaction specific membrane microdomains. You can have a modification of a gene expression which alters the membrane lipid composition. You can have a direct membrane lipid therapy, drug binding to a protein which alters its membrane binding affinity or that of other signaling proteins. You can have a regulation of enzyme activity to alter the membrane lipid levels. There is also a modification of membrane structure sometimes. So, and there are a lot of disease which has been which are under the trial and which can be treated by using these membrane lipid therapy. For example, it plays a very important role in oncology treatment, metabolic and cardiovascular diseases, and as well as in neurodegenerative disorders. Now, another major component of the biomembrane, as I mentioned earlier, it's proteins. What are those proteins which are present? What are the types of proteins which are present in the biomembranes? First, we will talk about the integral membrane proteins. Integral membrane proteins, they are also known as integral or intrinsic because they are deeply embedded in the membrane. So if you look at here, the structure, so these are the integral proteins which are embedded in the membrane. So if you have, they are exposed to the aqueous environment on both the sides of the membrane, and part of these proteins are in van der Waals contact with the hydrophobic region of the membrane. The major function of these proteins is they transport molecules across the membrane, for example, insulin, glycophorin, etc. Now you have peripheral membrane proteins, which are the extrinsic proteins. Sorry, they are located on the surface of the membrane. So these are the peripheral proteins. They are weakly bound to the membrane surface by ionic interactions or by hydrogen bonds which forms between the membrane polar heads and proteins. Example, cytoskeleton. They can also be recovered directly without disrupting the membrane. Now, another type of protein is transmembrane proteins. They basically, if you look at here, these are the transmembrane proteins. They span the whole breadth of the membrane. If you look at in the structure, they are coming here and they are here as well. So if you look at the difference between integral and the transmembrane, they are completely emitted, but they span the whole breadth of the membrane. So the hydrophobic side chains of the amino acids are embedded in the hydrophobic central core of the membrane. An example of these proteins are, they basically serve as receptors for hormones, tissue-specific antigens, growth factors, neurotransmitters, etc. Now, so there's a combination of lipids and proteins, and there is a model which is called fluid mosaic model. That was this model of membrane structure. It was proposed by Singer and Nicholson in 1972, and it was fun. If you look at the structure, you can see it looks like a ice box which are floating in the seawater. So this is the aqueous structure, and you have a lipid bilayer. These are the integral proteins, peripheral proteins. Then you have a oligosaccharides which are attached to those membranes. Now, uh, some membrane proteins and individual phospholipids can move sideways. Basically, they can move laterally in each layer, and there are range of different proteins through the phospholipid layer or resting on the surface gives this mosaic appearance. So what happens here is, there's a in the structure, what happens is there are translational diffusion happens. So you have a integral proteins here which basically undergo the rapid distribution in the plane of the membrane, which is quite rapid for a molecule, phospholipid molecule within the membrane. So one molecule of phospholipid can move several micrometers in seconds, and basically there is a phase changes, and this mosaic appearance structure look like this is the membrane structure. Now, the fluidity of the membrane is highly dependent upon the lipid composition of the membrane, as you have already discussed earlier. And that basically helps, and the setting up of this model, which is known as a fluid mosaic model. Now, so the same integral proteins and the peripheral proteins, as we have already discussed earlier, they are present in membrane of nearly all vertebrates. The nature and the function of integral and peripheral proteins, they require very special attention, as because they're present in the cell membrane of nearly, as I mentioned, all vertebrate erythrocytes. We will talk about here in detail about the red cell membrane structural characteristics, in which we will talk about the glycophorin integral protein, band protein. So glycophorin integral protein, if you look at here the structure, and this is the structure for the glycophorin integral protein, which is having an N terminal and a C terminal on the opposite side. So these are basically the glycoproteins, which contains 60% carbohydrate by weight, in which the oligos are bound to glycophorin, which are linked to serine, threonine, and asparagine residues. Red blood cell membrane contains about 6 into 10 to the power 5 glycophorin molecules. The, the protein, the chain of the glycophorin contains 1 amino acid residues within the non-polar hydrocarbon phase of the phospholipid bilayer, as you can see here, and they are tightly associated with the phospholipid and cholesterol. 23 amino acid sequence has an alpha helical conformation, and this is basically the example of the glycophorin integral proteins are M and blood group antigens. They also act as a sites through which influenza virus becomes attached to the red blood cells. Now the another protein is band 3 integral protein. This is a structure for the band 3 protein. And this is the another major integral protein found in red cell membrane. It is basically a dimeric having a molecular weight of 93,000 Dalton. The polypeptide chain of the dimer is thought to traverse the membrane about a dozen times. If you look at here this structure carefully, the number is going again and again in this lipid bilayer. So both the C and N terminal, if you look at here C and N terminal, they are facing off on, they are on the cytosolic side of the membrane, and the N-terminal residues, they extend into the cytosol and interact with components of the cytoskeleton. They plays a very important role in the function of red blood cells. As red blood cells flow through the capillaries of the lungs, they exchange biocarbonate ions, which are produced by the reaction of water and carbon dioxide, for exchange of chloride ions. So this exchange occurs by way of channel in band 3 protein only, which forms a pore through the membrane. Thus band 3 protein plays a very important role in the membrane transport protein. Now we have the inner face of the red blood cells membrane, which is laced with a network of proteins called cytoskeletons that stabilizes the membrane and is responsible for the biconcave shape of the cells. Basically, those are the peripheral proteins which provides the stability to the red blood cells. The special peripheral membrane proteins they participate in the stability of the red cells are spectrin, actin, and chirin. Spectrin basically consists of alpha and beta chain, which are of molecular weight of 240,000 and 220,000 Dalton. They are fibrous proteins. They are linked through short chains of actin molecules and band 4-1 proteins to form alpha 2 beta 2 tetramers. The polypeptide chains are thought to coil around each other. Given an alpha beta dimer, they are 100 nanometer long and they are 5 nanometer in width. The another is the actin, which is a cytoskeleton component, and erythrocyte and RBCs contain 5 into 10 to the power 5 actin molecules. They basically filaments, they polymerize. The 20 actin molecules polymerize to each other and form a very short actin filaments. Another is ankyrin, which forms the red blood cell's skeleton, but none of these proteins attach directly to the membrane. They helps in the attachment of a network of spectrin, actin, and band 4-1 protein to the membrane. And chirin has a molecular weight of 200,000 Dalton. The protein has two domains. Then one binds to spectrin and the other to the N-terminal region of the band 3 protein, which we have already talked about, that extends into the cytoskeleton. Now there are certain clinical aspects which are associated with when there is a defect in this peripheral or integral protein. So what happens is, so basically there are hereditary spherocytosis and elliptocytosis. These are the disease which happen. They are basically inherited genetic abnormalities of red blood cells, in which red cells occurs in the abnormal shape. It becomes very fragile. They have a very shorter lifespan than the normal erythrocytes, in which what happens is sometimes there's a band 4-1 protein is absent. There is a mutation happens in the genes which codes for the membrane protein. So it results into the increased hemolysis, anemia, and it also can lead to a jaundice. Now we'll talk about the function of biomembranes. They play a major role in transport systems. There are different kinds of transport systems which are listed here. The first one is ion channels. So the essential role of basically biomembrane is to allow the movement of all compounds necessary for the normal function of a cell across the membrane barrier. And these compounds include a vast array of substances like sugars, fatty acids, steroids, cations, and anions. To mention few, these compounds must enter and leave the cells in an orderly manner for normal functioning of the cells. So we have specific channels for conductive channels, cation conductive channel, which are negatively charged, And within those channels we have an average diameter of certain nanometer. So for example, if you talk about first ion channels, these are the transmembrane channels. These are very specific and highly selective for sodium, potassium, chloride and calcium. They are well regulated by the presence of gates, and there are two main types of gated ion channel, which are ligand gated and the voltage gated channel. So in case of ligand gated channel, what happens is a specific molecule, it binds to a receptor and it opens up the channel. For example, acetylcholine receptor. Acetylcholine acts as a ligand which goes and binds to the acetylcholine receptor, and in the postsynaptic membrane, so there is a what happens is when this binding happens, there's a acetylcholine released from the presynaptic region which binds with the binding site of the presynaptic region, which triggers the channel opening and allows the influx of sodium ion. Then we have ionophores. Ionophores are basically, they are very small organic molecules. There's a shuttle for the ions movement across the membrane, and there are two main types of ionophores. One is mobile ion carriers, like for example valinomycin, and another channel formers is like bramicidine. Now we have water channels. Water channels basically, they compose of tetrameric transmembrane proteins called aquaporins. There's a movement of water in cells of the RBCs and collected vacuoles of the kidney. These are the functions. There's actually a cause of lot of nephrogenic diabetes insipidus. This happens when there is defect in water channels. Now we have a gap junctions. Gap junctions are basically, they are composed of protein which are known as connection. It helps in the intracellular communication within the cells. They mediate and regulate the ions and small molecules passage. So these are the major functions of the gap junctions. What happens is, these are basically, certain cells develop specialized regions on their membrane for intracellular communication, which are present in very close proximity. So what they do is, they there's a passage of up to 1200 2000 molecular weight of molecule through a very narrow hydrophilic pore connecting the cytosol of the adjacent cell. These are the major function of the gap junction. Now, what are the types of transport mechanism? We know there are certain channels that the transport happens, but how it happens? So there are three different types. There are three important actually mechanisms through which the transport occurs. The one is passive or simple diffusion. Second is facilitated diffusion, and the third is active transport. And there are a lot of factors which basically affects these transports. There could be a concentration gradient, basically the movement of solutes from higher to lower or lower to higher concentration. Electric potential, there's a movement of solvent inside the cell which basically has a negative charge, and there is a hydrostatic pressure gradient. There's a temperature, permeability coefficient. These are the factors which basically affects the transport mechanism. Now, if you see here, what are the differences and what are exactly the passive or simple or facilitated diffusion? Passive, it depends on the concentration gradient. Facilitated, it's similar to passive. In passive, this process does not require any carrier protein or energy. However, facilitated, it requires a carrier or transport protein, and but it cannot, and it can operate bidirectionally. However, passive is always unidirectional. The rate of diffusion in facilitated is faster because there's attachment of a carrier or transport protein which facilitates the process as compared to the passive diffusion. This is the equation generally used for the passive or simple diffusion. Here we need to have an initial rate, solute, concentration gradient, the thickness of the membrane through which the solute is passing, and a diffusion coefficient, that is the how much is the area which is covered in how much time. The examples of passive or simple diffusions are water, gases, and pentose sugars. The examples of facilitated are D-fructose is absorbed from intestine specifically by facilitated diffusion. The another transport is active transport. It takes place against a concentration and electrical gradient, thus it requires energy. About 40% of the total energy requirement in our cell, it is utilized for this transport system. It basically requires the mediation of a very specific carrier or transport proteins. So what are basically the active transport systems? So there are basically two, which are further divided. And the one is the uniport system. The best example for the uniport system is glucose transport, in which what happens is there's a transport of a single solute molecules through the membrane. Then we have a code. So what happens is in this, we have another is called co-transport. We have a symport and antiport. So in symport, there is a transporter which carries the two solutes in same direction across the membrane. However, in antiport, there are the two solutes which or ions which are transported simultaneously in opposite direction. So in case of symport, the example is transport of a lot of amino acid across the intestinal lining in the human gut. Antiport, for example, chloride and bicarbonate ion exchange happens in lungs or in the red blood cells. The another examples are D-glucose, D-galactose, amino acid transport, etc. Now, how this transport of molecules occur, whether it's active transport, how it basically happens. So the mechanism of transport of macromolecules such as proteins, hormones, low density lipoproteins, viruses, or immunoglobulins, it takes place across the membrane by two independent mechanisms, which are exocytosis and endocytosis. So in this exocytosis, if you look at here, what happens is it basically involves the contact of two inside surfaces, which is a cytoplasmic side monolayers. It is also known as reverse pinocytosis. So what happens is most cells release macromolecules to the exterior by using this process called exocytosis. This process is also involved in membrane remodeling when the components synthesized in the Golgi, which are carried in the vesicle to the plasma membrane. The movement of vesicle is basically carried out by uh, cytoplasmic contractile elements in the microtubular system. So what happens is the mechanism is the inner membrane of the vesicle fuse with the outer membrane, uh, which the cytoplasmic side of vesicle fuses with the cytoplasmic side of plasma membrane. So this process is basically triggered by a local and transient change in calcium concentration. Similarly, endocytosis also requires the calcium concentration. It involves the contact of two outer layer instead of two inside layers, and it requires a lot of energy which is derived from the ATP hydrolysis. So what happens is all eukaryotic cells, they are continuously injecting parts of their plasma membrane. Endocytic vesicles are formed when segments of plasma membrane, if you look at here, invaginates, enclosing a minute uh, volume of extracellular fluid and its contents. The vesicle then pinches off here as the fusion of plasma membrane. This seals the neck of the vesicle at the original site of the invagination. Then the vesicle fuses with the other membrane structure and thus transports its contents to the other cellular compartment. Most endocytic vesicles, they fuse with primary lysosomes to form the secondary lysosomes, which contains hydrolytic enzymes and are therefore specialized organelles for intracellular disposal. The vesicular contents are digested, liberating simple sugars, amino acid, etc., which diffuse out of the vesicles to be reutilized in the cytoplasm. Now, there are further um, types of endocytosis. It could be a phagocytosis or pinocytosis. Pinocytosis is having a different type, which is can be a fluid phase or a receptor mediated absorptive pinocytosis. Now first we will talk about the phagocytosis. Phagocytosis means it's the word to eat. It's basically the engulfment of all large particles, for example virus, bacteria, cells, cell debris, by granulocytes and macrophages. What happens is they extend the pseudopodia. These pseudopodia are basically the false food and surround the particles to form phagosomes, which later fuse with the lysosome and result in the formation of phagolysosomes, in which there is a pH change and the particle starts getting digested. This biochemical mechanism which takes place through these is respiratory burst, oxygen consumption is increased and lead to formation of a lot of superoxide ion O2. Now there's a pinocytosis. The pinocytosis is basically it's a cellular uptake of fluid and fluid content. It's basically a non-selective process. So what happens is there's a uptake of a solute by formation of small vesicles, which is simply proportionate to its concentration in the surrounding extracellular fluid. Now you have a receptor-mediated absorptive pinocytosis. It happens because of the coated vesicles in the endosomes. So there's a transport of macromolecule from the exterior of its interior. So, so what happens is what happens exactly is the external surface of the plasma membrane, they're mostly covered with a lot of receptors and characteristic coated pits. Cell surfaces are rich in receptor proteins that can combine with the macromolecule, for example, the ligands, which are bound to the membrane. So these coated vesicles or coated, they are basically 100 nanometer in diameter and they have a very characteristic brittle coat uh, on their surface. The vesicles are basically covered with an unusual peripheral protein called clathrin. Now, this protein dynamine, which binds and hydrolyzes the GTP and is necessary for the pinching off of the clathrin coated vesicle from the cell surface. This is the figure which represents the receptor mediated absorptive pinocyte. This is a ligand. This is the membrane which have receptors. It results in the formation of a coated pit. Then you have a clathrin. And there's another clathrin subunit. It results in the formation of a endosomes. And there's a pH change happens, which basically maintains all the balances all these processes. And all the receptors with ligands, they move laterally into these coated pits. And so they're basically pinched off and they're internalized into all the vesicles. The best example of this receptor-mediated absorptive pinocytosis is the low-density lipoproteins molecule which bound to the receptors and are internalized by means of coated pits. So what happens is, if you talk about in detail, these are the periphery of the cell's interior, which are structure called endosomes. If you look at here, they have a diameter of approximately 0.3 to 1 micrometer. The internalized coated vesicle fuse with endosome, as I've already mentioned, and they discharge their molecules into the interior of the endosomes. Now here there's a low pH which breaks the linkage between the receptor molecule with a simultaneous release of a lot of clathrin, macromolecules, free receptors, membrane fragments, and most of them which recycle back to the plasma membrane to replenish further the population of receptors and the coated pits as well. However, the sum of the macromolecule, they continue to the endosomes and then move and by the help of microtubules further interior of the cells, where they again fuse with the lysosome or become associated with the vesicles derived from the, for example, the Golgi apparatus. Now, what are the clinical aspects for if there is a transport system dysfunction? So that these are the list of disease or disorder which can happen. For example, chronic granulomatous disease. It is implicated due to defective phagocytosis and respiratory burst. It is characterized by recurrent infections and widespread granuloma formation in various tissues like lungs, lymph nodes, skin, etc. There is a receptor-mediated endocytosis dysfunction. There are a lot of viruses which are responsible for many diseases, which hepatitis virus affecting liver cells, poliovirus affecting motor neurons, AIDS affecting T cells, and also there's a iron toxicity which occurs with excessive uptake due to endocytosis. Now we have a cystic fibrosis which we've already discussed earlier. So there's a defect in epithelial chloride transport which uh, affect the different organs, including the lung, intestine, pancreas, kidney, and the sweat glands. So this is basically a recessive genetic disorder. It's chronic bacterial infection of the respiratory tract and sinuses. There is a protein called CFTR, which is a cyclic AMP dependent regulatory protein for chloride channel. There is a abnormality happen in the membrane chloride permeability, which results in the increase of viscosity of the most of the bodily secretions, which leads to this disorder. Now we'll talk about the third component of the biomembrane, which is not a major component, but approximately 5% of the weight of the cell membrane is carbohydrate, which is present in the form of glycolipids and glycoproteins. So if you look at here the structure, these are the carbohydrate moieties which are attached to the integral proteins. Presence on the membrane outer surface has been shown with the use of plant lectins, protein agglutinations that bind specific glucosyl residues. Example, convaline A, which binds the alpha glucosyl and alpha manosyl residues. Carbohydrate chains of many glycoproteins show structural variation from one molecule to another molecule. Basic function of these carbohydrates in biomembrane is recognition of cell. It protects the microvilli in the gut. They plays a very important role in the food digestion, and also they plays a very important role in the biofilm formation. Biofilm formation plays a very important role in the virulence and the growth of lot of bacteria, especially in the mycobacterial species. Now these are the references that can be thoroughly studied for the better understanding of this section. Basically the biological membranes and the structure and function. Follow us on ITLS Academy, and thank you.